am pleased to introduce today's speaker, Christine Sitt, who is our 2022 Vaughn Jordan intern. Christine grew up in the Atlanta area and graduated from Cornell University in 2021 with a bachelor's degree in environment and sustainability and a minor in soil science. After graduating, she worked in El Yunque National Forest in Puerto Rico before arriving at Archbold this past February. Her independent research project, which she'll be sharing with us today, focuses on plant traits and specifically how they shift along environmental gradients. So for her project, Christine became a real expert at researching new lab protocols and adapting them to our scrub plants in order to measure a whole bunch of traits, which we'll hear about today. And I can honestly say that I learned quite a bit about plant traits just from working with Christine on this project. In addition to her project, she's been a wonderful person to have in the plant ecology program, and in particular has done a tremendous job of managing our fungal collections for the last few months, and also taking very nice macro photos of all of our plants, some of which will be featured today. And then finally, she's been an important presence at the station. She served on the DEI committee, assisted with several summer camps, outreach events, virtual field trips, I won't announce a new job for her, but she has one that she is in the hiring pipeline for, but it will remain secret for now. And with that, I'll turn things over to you, Christine.
All right, so before I begin, some of you will notice that in the top right, there's a little box that says put video here. This is for people joining on Zoom. If you have the seminar in full screen mode, you'll see this video of my face. And if you just take that and move it to the top right, I made sure that the top right corner will be will be blocking minimal things in my presentation. All right, so that's my announcement. Here's the start. 大家好，我叫薛叶明，英文名是 Christine Sitt。很多人认为，科学研究只存在英语语境之中，但其他世界上大多数人的母语并非英语。 在我意识到这一点之后，我希望能用中文来介绍我的科研项目。非常感谢大家的倾听。那我现在开始用英文了。Hello, my name is Christine Sitt. My Mandarin name is Shui Min. In Cantonese, it is Si Man. Science is a thing that is usually recognized only in English, even though the majority of the world doesn't actually speak English as a first language. I wanted to do what I could to recognize this in my presentation, so thank you for your support in raising awareness of this issue. Laura, I just want to check if you had the captions turned on. Sweet. Okay, now let's talk about plants. How to survive in stress, the Florida scrub plant edition. How does a plant survive stress anyway? Well, let's look at the different stresses that plants will face in the Florida scrub. There are many stressors in our unique environment: acidic soils, low nutrients, intense sunlight, intense heat. I focused my project on the water stress across 3 different habitats at different elevations. So the 3 habitats are flatwoods, scrubby flatwoods, and rosemary scrub. Rosemary scrub is the habitat that is the most water stressed. It is at the highest elevation. And then flatwoods is at the lowest elevation, and it's typically near seasonal ponds and gets flooded really easily. For example, the past 2 weeks since Hurricane Ian, the flatwoods that I've visited on station property have been really flooded and had a lot of standing water. For my particular project, the lowest elevation that I had was 37 meters, and the highest elevation that I had at the Rosemary Scrub was 47 meters. And the thing is that actually between my different sites, some of them only varied by a few meters between the Rosemary Scrub and the Flatwoods. So I think it really just shows how much of a difference there can be from the smallest change. So with all of the different stressors that plants face here, plants have developed different life strategies. I did that too soon, different life strategies to survive. So there are a lot of different life strategies that exist. An example might be the leaf economic spectrum, which is heavily studied, but I focus on the acquisitive conservative, AKA fast slow, growth gradient, and the collaboration gradient. I think the main key thing here is that with every strategy, there's always going to be a trade-off. So for the acquisitive conservative growth gradient, plants typically will try to conserve resources when they're building infrastructure. So that's leaves, their stems, their roots. So it's just kind of like how much energy, how much resources they're putting into each of these different plant organs. And so there's always going to be a trade-off, at least for this growth gradient, between having plant parts that are cheap and easy to build, but weak and not long-lasting, or ones that are expensive to build, but will last a lot longer and are generally sturdy. And so I think a good example for this here is, which building would you rather have to survive a hurricane? So on the left, we have the easy to build straw grass house that you can really acquire the grass from wherever, and it's gonna come up and be built really quickly. But on the right, you have the more expensive but durable wood building. And then there are some people like John Roebling, who built Archbold Biological Station, who can get a hurricane shelter that has thick foot cement walls to survive the hurricane. Most of us, however, would be satisfied with the sturdy wood structure in the middle. I think the main highlight, though, is that there are all these different options for building a house, but there isn't a right way to build a house. It really depends on the goal and the climate of what that you live in. And so the same thing is for plants. For plants, water-stressed plants, especially, they are expected to build expensive leaves that are thicker and sturdier. So the scrub rosemary on the left here, versus on the right, the Lyonia fruticosa is in less water-stressed habitats. And so it tends to build leaves that are cheaper and larger, but they're also weaker and they're going to lose a lot more water due to surface evaporation. And in addition to that, they're also more susceptible to damage. So which one is better? Again, it depends on the circumstances. Lyonia fruticosa prefers the lower elevation habitats, scrubby flatwoods and flatwoods. And so these are the ones that are a lot less water stressed, whereas rosemary is almost entirely in the aptly named rosemary scrub habitat. So it just really depends on the situation here. Moving on to the collaboration gradient, the focus was on the below ground plant traits because I think that roots are really interesting, and like leaves, they can be expensive or cheap to build. So on the left in this graphic, you'll see a plant that built really fine roots that can just reach far away resources, but these roots aren't going to last very long. Versus on the right side, there's another strategy of growing a big taproot, which is really sturdy. It'll last a long time, but it might not be able to reach all of the different resources in different parts of the soil. And so going on to an example that is inspired by a true hurricane story, something that plants will do is, as I said, they'll build these resource-seeking root networks themselves. In the case of the person who's sweating and sad, with the straw house, that's really easy to build. Versus the alternative is, say you have a resource like pie, and then you trade it with some friends and you say, hey, I'll give you some pie. You'll help me build this wooden structure here to survive the hurricane. You can come in it too. Or in the case of John Roebling, and you have many, many pies, you can build the aforementioned hurricane structure. So my project seeks to answer these questions. How do plant strategies shift in response to stress? And are there differences in strategies across the two families, the Eurycaceae heath family or the Fagaceae oak family? And I hope everyone pays attention in the next few minutes because there might be a quiz later. So to answer the questions, we go back to the plant strategies. And so on these two charts, most of the traits that I studied are written down, and I show how they're related to each life strategy. There is one trait that I didn't include, which is a leaf shape trait. It's called circularity, basically how close the leaf is to being a full circle in shape. And the reason why I didn't include it in these charts is because it doesn't really fit in with the growth strategies. It's really just more of a descriptor of the plants. So since I studied a lot of traits, it's too much to go into the specifics of each one. So just know that I use standardized protocol when sampling and measuring my specimens, or you can interrogate me afterwards. For the fast, slow growth gradient, this is the one that is the trade-off between building something that is flimsy, but easy and quick to build, not expensive, or building something that is sturdy and expensive, but will last a long time. So when plants are stressed, it's expected that they're going to grow smaller leaves in relation to their mass, so their resources, and then the leaves will be denser with the lowest surface area. And with the lower surface area, they'll have a lower stomatal density, meaning that they'll have less surface water loss through evapotranspiration and gas exchange. For the cooperation gradient, I want to go ahead and give credit to previous plant intern Alon Tran, who led the ectomycorrhizal study here at the plant lab at Archbold. Without her, I would not have had the opportunity to measure specific root length, and I definitely wouldn't have had any of the ectomycorrhizal root density data that I'd have. So for the collaboration gradient, there are the two strategies I mentioned earlier of doing it yourself, building these expansive resource-seeking networks yourself, or collaborating, trading pies, and really just kind of working together. And so it's expected that when plants are stressed, they're going to build these thicker roots, so less focus on seeking out all the resources themselves, but then they'll have fungi that they'll work together with to trade resources with, and then the fungi will be able to reach further and gather the resources. In total, from the 2 plant families and the 7 plant species, I looked at 260 individual plants, and it took a long time. The map to the left here shows my different study sites, which I spread all over the main Archbold property. At each site, I had the 3 different habitats: rosemary, scrub, scrubby flatwoods, and flatwoods. For my oak species, they were present in every single habitat. They're just everywhere. My Eurocaceae species, however, were more habitat limited. They were really limited to depending on the species, actually. So the 2 Lyonias and the Beharia were limited to flatwoods and scrubby flatwoods. However, the rosemary, as I mentioned earlier, is limited to rosemary scrub, if you couldn't figure it out. Now that we've covered what I studied, let's move into what I found. Most of my traits didn't really have consistent patterns, so I am focusing this presentation on those that did. For these 2 charts here, they are essentially almost the same trait. It's leaf dry matter content and twig dry matter content, so the amount of resources that plants are putting into their own growth for their leaves or their twigs. And so on the x-axis, it's the different types, the different species. So Lyonia lucida, Lyonia fruticosa, Beharia racemosa, these are my Eurocaceae. The Q other stuff, Q geminata, Inopina, Chapmania, these are my oaks. And so for each of these species, it shows the different measurements that they had by the different habitats. So for Lyonia lucida here, you can really see that for the twig dry matter content and the leaf dry matter content, as they go from being in the flatwoods to the scrubby flatwoods, so low stress to higher stress, they increase their dry matter content. So they're putting more resources into building these sturdy these sturdy plant infrastructure. And so also on the bottom in between the 2 graphs here, you can see what the different stars or pluses mean for how statistically significant they are. The Lyonia lucida dry matter content is the 1 star, so that's the P less than or equal to 0.05. And then the Quercus inopina, Quercus chapmania, for the graph on the right, the leaf dry matter content one, they are a bit more marginally significant with the plus sign. And so, yeah, as I already mentioned before, some of my species, the Eurocaceae, aren't really prevalent in all the 3 different habitats, which is why the plants on the left don't have the red bar, because they weren't in the rosemary scrub, because they don't really live there. Rarely they do, but not enough where I could really sample them from that. Okay, so another trait that I don't show here is stomata density. And the thing about the stomata density is that Quercus chapmanii, one of the oak species, It did significantly decrease its stomata density as it went from lower stress to higher stress environments. And this is expected. However, this only happened for the one species of the stomatal density trait data. So I didn't include it here, but I think it's something that is important to note. Therefore, for the above ground traits that I studied, there is evidence that plants do somewhat grow more conservatively. However, things get really interesting when looking below ground. The oaks are really interesting when looking at their root and ectomycorrhizal data. There's some evidence for conservative growth when they're stressed in the below ground trait data. It does happen. It's not strong, but it does happen. You can see this on the graph to the left, which is the specific root length. And specific root length is the example that I brought up earlier of taproot structure as the strategy versus expansive fine root networks. And so for this graph, it's all the oaks versus the specific root length. And so you can see that for each of the habitats, as they go from lower stress to higher stress habitats, scrubby flatwoods to rosemary scrub, the higher stress rosemary scrub specific root length is lower. And so it's not extremely significant, but there is evidence that it is there. For the graph on the right, this is where it shows that there's strong evidence that stress leads oak species to collaborate more. And so for this figure, it is about ectomycorrhizal tip density, so how much the fungi and oaks are really collaborating. So it's the same 3 oak species on the bottom, the same habitats, scrubby flatwoods and rosemary scrub, and then just kind of showing the difference of as the plants go from being in scrubby flatwoods to rosemary scrub, they increase how much they are collaborating with fungi. And all of them are statistically significant. So this means that as oak trees are in more stressed environments, they're going to spend less energy growing a ton of small roots that can reach a lot of different resources everywhere in the soil. And instead, they're going to trade resources with fungi and have the fungi form these resource-seeking networks. This is another way to see what I've already been saying. These charts here show the effect of stress on each of the traits and which traits are for which of the aforementioned life strategies. So the x-axis, which I don't think you can really see, but they're all the different traits that I studied. And then the y-axis is the impact that stress had on each of the traits. So blue is the fast, slow, or acquisitive conservative growth gradient. Red is the collaboration growth gradient. And then yellow is the weird leaf shape thing that I mentioned earlier that doesn't exactly fit into each of the life strategies. So they're just kind of a thing by themselves. Any graph that doesn't have a bar, it's because I didn't have specific trait data for that plant species. And so I want to note also that specifically for oaks, they are ectomycorrhizal associating, but the Eurocaceae species are not, which is why the top 3 graphs for the 2 Lyonias and Baharia don't have the root data. So as you can see with the different groups, the impact of stress on each of the different traits, it's really, really pronounced in the collaboration. They have 3 stars, so their p-value is less than or equal to 0.001. And the scale of the red bars is so much greater than the other traits. And so this really, really shows how much more of an impact there is on the collaboration gradient than any of the other traits. And so this really shows that oak species in more stressful environments will collaborate more with fungi, and then plants from both families will, in general, somewhat shift to a more conservative strategy. I hope everyone was paying attention because it's quiz time. Get your keyboards or your voices ready. So for the people in the room, I'll do a 5-second countdown. For people on Zoom, you can just kind of type in the chat, and then I am definitely going to broadcast it on display for everyone in the room. So for everyone in the room, do plants, I think I lost my place. Do plants, when they're more stressed, based on my results anyway, grow faster or slower? 5, 4, 3, 2, 1. Slower. Amazing. All right. And then when they're stressed, do they tend to collaborate more or build their own root infrastructure? 5, 4, 3. Wow, that's a lot of people. I think the Zoom crowd beat the in-person crowd. So go Zoom people. Yeah, I didn't even get to finish the countdown. They cheated. All right, so yes, the answer is they are going to collaborate more. So thank you to everyone who participated in the quiz, whether or not you got the chance to answer both questions. For the second question, are there differences in strategies across the two families, the Eurocaceae heath family or the Fagaceae oak family? And there really wasn't any evidence for strategies that differ, but I do think that it would be interesting to study further into plant traits and how they differ across more plant families, because I was only limited to the two families. And so speaking of interesting studies for the future, I think it would also be interesting to not only look across more plant families, but also in individual plants. So if they go, rather than comparing individuals in lower stress versus higher stress environments, I think it would be interesting to see how plants maybe might be able to shift as they go from lower to stressed environments in the same place. I think it would be really interesting to look at the relationship between soils and plant fungi interactions. I don't think anyone in the room can see, but I have a sticker on my laptop that says, "I heart soils." So if that gives any indication into where some of my interests lay. But yeah, so I think it would be really interesting to look at the vastly understudied below ground plant parts, so roots, but also other organisms and other interactions that go on there. And then something else that would be really interesting for the mycorrhizal aspect of this is if hyphae, so the little tiny root edges that are able to take in resources, I think it would be interesting to see if they transport water as well as they do nutrients like, say, phosphorus. So I want to give a big thank you to a lot of people, but my co-advisors, Erin and Greg, for their guidance, support, and their expertise. Members of the plant lab who started before me, who are always open to talking through project issues and answering any random question I had. Members of, that's the wrong order, my friend Fu Zishen, who helped me with the editing of my Mandarin introduction. Plant lab members who started after me, who make the plant lab continue to be such a welcoming and supportive environment. All past and present residential staff of 2022, at least the ones I knew, who provide such a vibrant community to come back to. The diversity work group for making Archbold a better place, and everyone else I haven't directly named because there are just way too many people. Are there any questions? I guess.
So I was curious with the cryptogamic, the soil crust communities, which my understanding could be wrong, generally a little bit more obvious in the higher, drier areas, and whether the mycorrhizal communities in those are really tapped into not just a collaboration, in terms of fungal, but also a sort of collaboration with these really quite complex cryptogamic soil crusts.
So the question was with cryptoamnic soil crust.
No, cryptogamic.
Cryptogamic
soil crust.
Soil crust, if mycorrhizae are going to be forming different associations like with plants in the different habitats versus what's going on with different soil crusts. Is that correct?
I'm curious whether the soil crust, the ectomycorrhizal fungi, and I don't know this, I'm definitely not a soils person, but I'm just curious because they're generally in those higher, drier areas, are they actually providing an interface between some of the valuable nitrogen and other sources in the soil crust and the, the ectomycorrhizal plants and the oaks?
So I unfortunately do not have any detailed answer because soils here are vastly understudied. But it's just like in general, everything is really stressful in all the different types of soils. So the quick answer is, I don't know. But I think it would be interesting to look further into. Yes.
So great presentation, obviously very plant focused from your perspective. What about the potential impacts of the stress on the fungi? So the oaks, right, are potentially collaborating much more in the stressful environments because it's good for the oaks. Is it also good for the fungi? Are they not present in the less stressful spreading flatwoods? Do we know anything about how they respond to these different gradients?
Mycorrhizal systems are just an entire other thing to really go into. And admittedly, I don't know a lot about it. But the kind of thought, or not thought, but like what happens is, so plants will be trading resources with the fungi. So the fungi will get, is it carbon or nitrogen? And then, so they'll be trading resources. And so the fungi will be the ones getting specific resources and then giving them to plants for other specific resources that the fungi are not really able to get. Did that answer that question?
Yeah, I'm just sort of looking to speculate on whether there's something different that the fungi may or may not be able to do in one place or the other. But yes, you explained it, explained like the village.
Okay.
Good, thanks.
Okay. There are some online questions. Do I just?
I can read them out loud.
Okay.
So Iris asked, such a cool project, Christine. What did you find to be the most challenging part of your work?
Oh my gosh. Thanks, Iris. I think it was really difficult because I had so many different parts to my project. I had 6 or 7 different traits and like 7 different plant species. And so it was really difficult for me figuring out how to scale everything and knowing when to scale down, because throughout everything, I was like constantly scaling down and focusing and reframing what my project was.
Abe asked, I think you may have the species wrong. How might Lyonia pharynginia traits compared to Lyonia fruticosa traits? The former seems more restricted to stressful xeric habitats.
So you think that's supposed to be lucida and fruticosa?
Or misread.
I didn't, well, Abe, I didn't really study pharynginia, but I did study lucida and fruticosa. And so I think one thing that was showing up in my data, if I can go back to it without it taking too long, is that for the dry matter content, lucida, which is the leftmost, and then fruticosa, which is the one to the right a little bit more, they have different dry matter contents, which I thought was interesting because dry matter content, at least how it's expressed in this chart here, is the ratio of how much the mass of the plant compares to like the mass of the dry plant, sorry. So like the solid matters, how much that compares to when it's fully fresh. So when it has water and it's not oven dry. And so at least for dry matter content, they have that already difference because, so I know you know this because you have been here for decades, but lucida versus fruticosa, Lyonia fruticosa is a plant that is very like fuzzy. It has like hairs on the leaves and it's pretty round sometimes. And then lucida is a lot more flat and smooth. And so something that was actually different between the two of them with one of the traits I was studying was stomatal density. And I could very easily get stomatal imprints off of Lyonia lucida. Lyonia fruticosa, I couldn't get anything off of them. I couldn't see anything. I just saw imprints of hair.
So I'm curious. So what is the purpose of hair on plants? Well, no, I'm just actually curious how you mentioned it. If that just increases like surface area, or if it's something else. Or...
So the question was, what is the purpose of hairs on plants? Because I just talked about hairs on plants. And so I think what it is that they have a couple different things that they can do. For one thing, yes, you mentioned they increase surface area. And so that can be good for like holding on to water vapor. Another thing that they can do, depending on how fuzzy or hairy they are, is it can help with herbivory. So it can make it more difficult for insects or animals to really eat them, because I don't think anyone really wants to eat a cotton ball. So those are a couple examples.
Could you ever remove the hair?
Could I have removed the hairs? If I just put a little bit more willpower into it. So one of the things that I did in preparing all of my samples for stomatal density is that I was actually washing the leaves, the backs of the leaves, before I took the imprints with nail polish, by the way, if you ever wanted to find a fun use for nail polish. So no, I couldn't have done any better.
Why do you think the traits didn't show consistent patterns? So you didn't see a lot of significant effects of the stress gradient?
So the question was, why were there not consistent, why was there not consistent data across the different traits? I think it could be a lot of reasons. One could just be there are way too many factors that I was really just not measuring or aware of. Another thing could be that, when I picked my sites, I picked 10 sites that were across all of the main Archbold station. And so I was really generalizing the sites that I had. I wasn't going to specific ones that were all really similar. And so in that way, there's going to be a lot of variation just kind of existing. So I think Akeem has been raising his hand for a while.
What made you decide on passages for this study? Like what made them best for the study? And kind of a follow-up, like, or not really a question, but I just thought it would be interesting if there was any difference in the differences, if it was like grass species or sedge species or forests instead of shrubs.
So the question is, how did I choose the plant species? And then if there might be any differences over what the types of species were?
Or like if you didn't choose shrubs to do the study, like what do you think would happen with like grasses or sedges or forests?
If I didn't choose the species, these specific species, and chose other ones, what do I think would happen for grasses or forbs? So how I picked the species was I went up to my advisor, Erin, here. And I was like, hey, Erin, I want to look at the most common plants that are here. And then we talked through what would be best for the traits I was considering at the time anyway. And so those were the shrubby species, the most common ones. And so the second part of the question, which is what would have happened if I had looked at forbs or grasses instead, I know that for stomata, at least, it's like a really weird trait to measure. And so I think I would have had different difficulties with measuring that one. For dry matter content, I kind of expect that it would be lower because I think grasses in general may be less dense than like woody plants like shrubs. So different expectations.
I don't have any questions, but so you were saying how with the oaks at these higher stress levels or other plant species that basically there's more collaboration happening with the fungi. And so what do you think is happening? Could there be an effect of, I don't know if fungi form associations between multiple plants, or I don't know if anybody else here knows that basically, or if it's based between specific fungi species associated with specific plant species. So like a density gradient of having a lot of one certain species in one area is going to cause more collaboration, or if there's no effect at all. I don't know if that, am I explaining this well enough?
I got a little lost. Are you talking about density of the species in a specific area and how that
Yes,
impacts
that
the
affects,
roots,
like,
the fungi?
collaboration gradient of the fungi.
I think that would be an interesting further study to do. Dustin.
That was a great talk, and it was reminding me of a summer camp and how great summer camp activities were with your call and response there. My question is about the in the relationship between the plants and the fungi. Is it the plant that is saying no most of the time? How come is it when the plant is stressed, it's like, hey, I want to be friends. And the fungi always wants to be friends and is just waiting for the plant to get stressed, like, yes, now we can be friends. What's stopping the other ones from having so many relationships with fungi?
So what's stopping the Ericaceae family from associating with fungi because the oaks are associating and wanting to be friends, but the Ericaceae are not? Is that the?
Well, because the oaks, when they were less stressed, only like didn't have as much relationship. So were they actively looking for more fungi relationships when they were stressed? And like, what stops them from not having that relationship all the time?
So one thing that happens with fungal associations with plants is that it actually can be costly. So it's like in my pie example, you know, you can trade with people or fungi to be able to build this infrastructure. What if you don't have any pie? Then you just can't offer anything. So then you can't form the associations.
That's a perfect answer.
You can also just wait because this was alongside of the project.
Right.
The data here is the colonized tips. So basically what that means is that the plant produced these tips on its roots, and then the fungus came and colonized it. And that's what was measured. So the plant is basically, you can think of it as like asking for the fungus by producing these tips, and then the fungus comes and colonizes them.
Cool.
Nova?
Yeah, so I'm a little bit curious. You took different sites at different elevations and you measured with different levels of moisture. But since Archibald does control burns, I was wondering if you factored burn times into any of your sites or if any of them were burned more recently. Might this be very different from somewhere nearby that does not have controlled burns? Do you have any kind of ideas? That's just one of those factors that is external to everything else.
So the question was focused on, because I spread all of my sites across all of Archbold, and we have different burn strategies and timings that different sites are burned, how that would have impacted the traits. Yes, okay. So yeah, I did look at burn data, and I did have some sites that were burned maybe, I think the lowest was like 2 years ago, and the longest was like 20 or 40 years. So there were definitely differences, at least when I was going through the sites, of how dense they were. Dense as in like the plants that were there, not the individual specific plant traits. I didn't really look at the data for this, which would be another confounding variable from the earlier question about why there is so much variability. So I didn't look at it, but it is something that a lot of people study here, is the impact of burns on different things.
I think we can take one more question. Julie?
What is your favorite plant that you've encountered in the scrub?
The last question is a fun one on what is my favorite plant. And I think I actually have a picture of it right here. And so it is Euphorbia rosescens scrub spurge, the lovely name of scrub spurge for whatever reason. And I find it fun because it has reproductive structures that can be male, female, or bisexual. And it's really interesting. And also just, it's my favorite plant demography project because it's kind of fun to go to the sites and be like, I found this seedling and there are so many, I'm just going to claim all of them before anyone else in the plant lab can.
All right. Well, let's all give Christine a hand.
Well, I don't know, I was just thinking thinking, this is the first time we've all been together in a room, and we always used to have a tradition of introducing new folks at Archbold, but people here have been here months to years who've never been introduced. And I just suggest we just go around the room quickly and just give our names and where we work.
Yeah, you want to come here and say it?
Yeah, sign off.
Okay, we're going to sign off on Zoom. Bye, everyone.
Everybody, we're gonna sign on. Thank you so much for watching today. Join us again in 2 weeks, October 27th, for a presentation by Susan Lohr and Stacy McMahon, Ranchland Conservation Legacy. See you.